the science you've been doing in the second half, right?
Yeah, that's right. I have about maybe 20 minutes, so as much discussion as we can do, I think would be great.
Okay, perfect. Sounds good. Sounds good. So I thought, maybe I can kick things off with some, general updates on things going on with folding at home, so that everyone's in the know, including, some about the companies that have gotten involved. So some things to fill everyone in on is, Joseph has undertaken a grand adventure, and he and his wife just in the last 2 weeks moved to Finland. His hours will have shifted, which will make it, different times where he'll be most accessible, but he'll still be doing the same work for folding at home. And we also in mid-September, a new developer named Shreya started full-time. So she is based in India, and her primary focus right now is working on the new version of the client software that we've been moving along in the background during these COVID times. And the idea there is that, right now the client software is actually, something like a half dozen different pieces of software communicating in different ways. And so really pulling together the core science part into a single piece of code that people will run in the background on their computers. And, so you'll have some control via the command line, but the primary way of interacting with it will be through a web portal that will be kind of like a Facebook profile for folding at home, where you can, see points over time, and, we'll be able to, because the UI will be running on our servers, we'll be able to update it more often and iterate more on that side. And so, things like adding in the COVID-19 option, which was a big pain in the butt, during the pandemic, will be straightforward. And then also on that side, now the latest version of Gromacs actually includes the ability to run entirely on GPUs. That makes it pretty simple to have a Gromacs GPU core, at least in theory. So Neha in my lab is testing it now, so that's a convenient option to have. In the new version of the client that Shreya is working on, we've also been including logic for having the capability to have hybrid work units that use the CPUs and GPUs. So hopefully that will just give us more flexibility in how we use these different pieces of software and what could hypothetically be done in future pieces of software. As far as membership and folding at home, we're onboarding 2 more labs right now. So one is Sonia Hansen, who had worked with John previously and now has a independent position at the Flatiron Institution in New York City. And the other is Antonia, or she goes by Tony May, who's been a longtime member of the Markov model community, and worked with Frank Noe, and now has a faculty position in Edinburgh. So Sonia is getting going now, and Tony will be getting involved in the coming months, and then Shuhui is getting more involved also, and has recently has now moved to the University of Wisconsin-Madison. So that's nice, because he's more in the he's in the U.S. time zone and not Hong Kong. So that'll make it easier for him and his group to be involved, and less scheduling, one less scheduling hurdle to complicate our lives. There's these working groups, and the science communication one was partly the inspiration, and that's been working out really awesome. So thanks to Sukrit and Raffel and Matt for spearheading that. And The PR one is getting underway. So Vince and Lucy are helping organize that, and Shuhui is going to be spinning up one that's more on the software side. So the idea is to update MSM Builder, since that's kind of fallen by the wayside, and maybe bring in some of the pieces that are useful from my lab's work with Inspara, or at least make them interoperable, and also to help keep track of some of the tools that are useful for folding at home. So my hope is that this can be a group of people who have some awareness of different tools people have made and can help us minimize some of the redundancy. So different tools for removing periodic boundary conditions, or things for doing adaptive sampling, or whatnot. And then with the membership starting to move into some of the things with companies, we have 2 companies that have gotten involved with as members of our consortium here, and that's Redesigned Sciences, largely based in New York City, and then Roivant, formerly Silicon Therapeutics, which is I think their home base is Boston, but I guess each of these companies is starting to open up multiple sites, so people are a little spread around. But I think this is an exciting opportunity because folding at home originally started with this very basic work on protein folding, and a lot of us have been moving more and more towards function and things related to drug design and other practical applications. And of course, something that we all, and especially our users, would love to see is real world impact in the form of contributing to the development of therapeutics. But of course, there's the valley of death between academic research and FDA approval of a new drug, where that's not an easy transition to make due to myriad things from manpower and resources and blah blah blah. And so it's always interesting to think about how can we have real world impact. And I think this is a cool opportunity where there's synergy, where the simulation methods and from Markov models and adaptive sampling and free energy calculations have matured to the point where they can be useful, but for real world applications. But they're definitely not turnkey, simple, press the green button and FDA submission ready compound for sending to the FDA pops out. And so this is a exciting time where some of the pharma companies, especially some of the earlier stage ones, have taken an interest in these tools, and are open to doing things out in the open as academic research, because they want to push these along and know that they're not giving away FDA approved drugs, for example. And so this is a really exciting opportunity to have some synergy. And I think one of the other components of this is that grad students, postdocs, PIs, were our primary job, if you will, is to have new ideas and new publications and do proof of principle stuff. And we all benefit from software that is done well, but that's a tall ask for someone to become expert scientist and professional software developer. Sorry, apologies for that. And so things are, there's a lot of academic software that leaves things to be wanting, and so I think another part of the synergy here is that I think the companies are far more able and willing to pay more professional software developers to contribute to things, and we can put our heads together on that too. So really cool opportunities for open science, and that's an important requirement. In part because we are dependent on the generosity of our volunteers. And, for many years, there's been subsets of our volunteers who get into conspiracy theories about how we're really working for pharma companies or whatever, right? And so of course we do want to be careful, because we wouldn't want to be trigger a mass exodus by actually doing IP generating things. But that's not even really possible, right? Because I think the fact that anyone can see what's going on their computer, the pharma companies, I think, would share our strong reluctance slash dread of putting things out unfolding at home, because it would be a nightmare for claiming IP, because it would be out there in the public. So anyway, so I think we're all on the same page there, but just so people are aware that we're not doing private IP drug discovery stuff, and to make sure we don't say things that would mislead any volunteers into thinking that is the case. But really we're working together on testing out what these methods are capable and what we can learn, and improving the tools so that they can be a part of forward motion on things related to drug discovery and other real world applications. So super exciting synergy. So Rafael in particular has been involved with two of these companies, and now is at Roivant, and they've been making exciting progress on some of the problems that they've been working to. So Rafael, do you want to add anything to what I said, or do you want to dive in with your presentation?
Yeah, so let me add a little bit to what you said. About the IP generation, we have a mechanism for real-time data sharing, right? So whenever what we know as a work unit comes back from anyone, there's people might be familiar. There's this next gen command in the project.xml, and we have a call to a Google bucket or for redesigns an AWS bucket, and that gets deposited there straight away. So for us, this is a mechanism of making sure that I really need to know that I can disclose something, and while we're not doing anything IP generating, I'm telling the entire world what we're working on. So it keeps us on our toes, and I think this has been a great idea. I'd like to know how many people, if anyone, is downloading that kind of data. It has a lot of data always, and you have certain capabilities to analyze it. But I think the fact that it's out there is great. Obviously, we've had the questions about right now the data sitting on buckets that requires a very small payment from anyone who would want to download it. And the new data for the preprint that I'll talk about, we've put on a bucket that we pay for. So we're going to see if we're worried about is that I still don't know for sure if this is possible, that kind of attack where someone would have a loop downloading files all over and drain our budget so that other people could use it. I don't think that's happened, and it's been out there for over a month. So if this goes well, then we will move all the data, the real-time sharing, to the bucket that we're paying for. Yeah, and so I thought this industrial collaboration would be a great idea, because what we have here is okay, I should say first of all what we do at Roivant. So like Greg said, Silicon Therapeutics was acquired by Redesigned Sciences. They didn't do any of their own drug discovery before, and they wanted someone to do it. So we're now called Roivant Discovery, and it's a team of both computational and experimental people. The lab is in Boston, and we have also free energy computational people over there. This team here in New York is new. It's called Advanced Simulations. And so we're finally doing a lot of things that the field has been wanting to do, that is use long timescale MD and enhanced sampling methods and so on, to try to be useful to medicinal chemistry campaigns. And it's an interesting process, and we're working out how simulation can help every day. So let me share my slides and tell you guys about.
Rafael, just before you, while you're setting up, let me just chime in to say that if anyone has any questions, please feel free to ask. I know this is being recorded and we'll post it publicly on YouTube as well, but If anyone has any questions about this that maybe even like internally oriented, that's like more on the folding at home side, this is primarily a forum for us to chat, so I can edit out those questions. So feel free to like ask questions that anyone wants, and if we feel like, oh, that question may be too internal, I can edit it out. So I just wanted to put that out there before we continue talking.
Yeah, and to add to that, my aim here is to get feedback on how we'd like this kind of data presented, because I think it's important that the stuff that happens in companies, there is regular updates to the entire world about what we do. So, some ideas are, I could write a blog post, I could record talks like this and drop them on YouTube. But I'm looking for what the right format is and what kind of data you'd like to see from these things. So please do interact with inside. All right, so let me in general talk a little bit more about what we're looking for from work with folding at home. Is what I call win-win open methodology development. So, as I always thought that one of the inefficiencies of this field is that, first of all, we tend to stick to one method and that we've learned somewhere. And so you have academic labs that do one thing well and they try to apply it to everything. And that obviously is not the optimal way to do things. So what we have here in this advanced simulation team is pretty much every single simulation technique and some static design techniques too that you can imagine. And we kind of try to battle it out, given a particular scientific problem, we're asking what is the most efficient way to do this. And for us, we can do the open stuff of folding at home, but ultimately the IP generating stuff, of course, has to happen on a cluster, which is of limited size and very busy actually. And we have the constraints of drug discovery cycle. So right now it's week long. So whatever method I deliver, it has to, within 3 to 4 days, it has to arrive at an answer, right? So we can do a lot of reference things of folding at home, or we do it over months, but ultimately we have to think about boiling it down to something quick, which happens within microseconds of simulation. So I'm going to show you a preprint on prediction of PROTAC protein-protein complexes. So first disclaimer, I came onto this paper very late, and so I'll show you just my contribution, but everything else and the gist of the paper is I had nothing to do with. But it's a fantastic method. They used weighted ensemble to, okay, first of all, let's talk about what PROTACs are. I'm sure many of you have heard about them. So there's a lot of excitement in pharma right now about this new modality of drug design, where instead of inhibiting something, you take a molecule that binds to your target of interest. It doesn't have to be such a good binder even. And then you link it to a second molecule that binds to a ligase. That ligase ubiquitinates lysines on your protein of interest, and that sends the protein to degradation. So the advantages of this, why people are excited, are one, you don't need nanomolar binders anymore. But second, they're also catalytic, right? Your molecule doesn't have to sit on that protein forever. It just has to go the once, it has to, and then this is where this protein-protein complex comes in. It has to create a productive complex where a reaction on a particular lysine happens. And then it can go away, right? Once the protein is degraded, you've freed the molecule. So it also removes a lot of the typical pharmacology worries about permeation and so on. But the problem with these protein-protein complexes is they obviously haven't evolved to actually be in touch, right? So I'll show you some movies. You'll see they're pretty, let's call them floppy. They're really mostly held together by this small molecule in there, and it kind of bouncing around on the surface. So you have many possible conformations, and you need to find one that, or many, that are capable of doing this reaction. And we have to sit in that conformation for long enough. And, honestly, we have no idea about anything when it comes to these things. There's some data that says it's better to have a very stable complex, sits there for a long time, eventually finds a conformation. There's some other conflicting data that says, no, actually, it's better to have it dance all over the surface and then by chance or sometimes find a conformation. No idea. So, the big game for us here is trying to go for a free energy landscape of protein-protein complexes like this to some kind of QSAR model of, first of all, binary questions like, is this molecule going to be that creates this kind of free energy landscape of these protein-protein interactions a good PROTAC or not, and then maybe trying to quantify it in some way.
Sorry, Rafa, what was it you said you had no idea about? Is it essentially like whether flexibility is okay in the molecule or if it needs to be?
Yeah, the big question of if I give you a free, even if I had a method to deliver a free energy landscape, and I'm sure about it,
yeah.
We don't understand what that means for degradation efficiency.
Got it. Mm-hmm.
So, my job is to deliver these free energy landscapes, and that's the stage that we're at. And then eventually, if we have some experimental validation of them in particular, we're going to try to answer the question of, okay, let's try to build a model.
Okay. So things like, does the orientation matter of the two proteins, for example, or?
Right. Yeah. Because you look at, we're also simulating these. This is also following home this whole Culian ligase complex, which actually, so you have this ubiquitination, and then 10 different things have to come together for the actual degradation process to start. And distances there for the ubiquitination are huge, like tens of angstroms normally in the crystal structures. And we really have to push it with some enhanced sampling to try to find conformations that get close. But, you look at it first without any pushing and you're like, how does this ever happen? But, there's also the point of we're not looking at the biologically relevant timescales, really. It has minutes there and sitting the cell to happen. So who knows?
All right, what is it that you aren't sure you would ever capture? There's a conformational change, or?
No, this is really like a thing bending, when these two proteins have to come together for
okay, got it.
lysine, where it's going to be ubiquitinated with a protein that ubiquitinates. The starting position is very, very far away.
Okay, got it. Mm-hmm.
Okay, but back to this. So this is also a very interesting story for what many of you probably be familiar from the mutation point of view, where, so what this figure is showing here, the first part B, we have, look at how few interactions there are in this protein complex, right? So again, it didn't evolve to be together. It's not like a nice looking key thing. It's just sitting there. But it could be crystallized. So it does sit in this one conformation of the crystal. The second part, we have 3 different protein molecules there that I can tell you functionally behave very differently. The crystal structure is pretty much the same. So you have the same situation with protein mutation happens very often. Crystal structure is the same. Thing does very different things. So clearly there has to be, well, first of all, one structure is not enough if we want to understand anything about how these proteins behave. And maybe dynamics are important and different between these different protein molecules, and indeed they are. So we're generating a lot of hydrogen deuterium exchange data. And so here you're looking at 2 complexes with 2 different molecules, so those I showed you. So the blue shading on the proteins is protection. There's less exchange of backbone amide hydrogens to deuterium. And maybe one of the most striking things to notice, what happens to my cursor? But look at the tip of that protein on the bottom left on in the top view. There's some allosteric kind of coupling with the interface in the right one, but not in the left one. While the right one has much less protection on the interface compared to the left. So many structures here that we need to understand, and maybe there's something interesting kinetically too. Yeah, so the big game is, can you generate a free energy landscape of this? and understand what that means. First of all, let me show you that indeed, yes, we can see many of these structures and the complexes are pretty floppy, if you like. These are snapshots from enhanced sampling, Hamiltonian replica exchange, MD. Zach, who made this figure, has chosen just visually what he thought after doing PCA and clustering of the HRMD data, the structures that he thought visually matched the HDX most. And the caveat here obviously is that the HDX comes as an ensemble property, comes from everything. And so we're kind of playing this game of still trying to satisfy our desire of looking at static structure. So you have the top structures or second most populated structure that seem to fit the data most for these different protags on top, and at the bottom there is the whole ensemble, just to show you the range of motion of the thing that's moving there is the ligase on top of the target protein. Okay, so what I'm trying to answer here today is comparing simulation methods and comparing data analysis methods. So let's first talk about comparing simulation methods. This is a movie from HRMD. So in HRMD, you run multiple replicas. Traditionally, you might know replica exchange where you change temperatures. Here you change Hamiltonians of a given part of the system. And the main point I want to make here is this is a movie in a completely random order, right? Yeah, we could have maybe done some RMSD clustering and plotted along RMSD, but ultimately you have lost kinetics in an enhanced sampling method. And a question that I can't answer, but I'm trying to answer, and I think this data set that we're sharing here is going to be great for, is this enough for what we're trying to do? Can I just give you an ensemble of structures with no arrow of time information between them? And is that enough for this kind of QSAR understanding of what these complexes do? Maybe, maybe not, right? And so this is what a power following home comes. We would never be able to generate an unbiased MD data set. It's only following home and D shot that is capable of doing something like this. And so what we did was we took the snapshots from this enhanced sampling simulation and we seeded simulations of following home to generate, we call a reference data set, right? And so now we have a 6 millisecond data set that we can ask any of these questions with. And so let me show you the very first view of this data set. So obviously it's 6 milliseconds and as always takes a lot of time. But first of all, I also want to point out that kinetics are gone from enhanced sampling. So forget about your TICA. You can do PCA, right? And so this is a plot of the PC0 and PC1, the two most variant principal components of this movie that I was just showing you. The red cross is the crystal structure for this particular complex, right? And so think about what, there's always problems with 2D projections, but ultimately, what are we to do? We try to get some intuition from these. So what is this telling? Well, it's kind of telling you that the crystal structure is actually really far away from the force field minima. It does appear like it is the PC0 that really describes the difference between the crystal structure and the minima. Okay, so let's compare this now. This is TICA plot now from the exercise I described. We call it enhanced seeding, right? We've taken 100 seeds. So in that PCA space we were looking at, we cluster into 100 states and take those as seeds and following at home and generate about 10,000 trajectories total. Excuse me, for 6 milliseconds. So that took like a month and a half, even for, now this thing's pretty big, like 100,000 atoms. So just to give you an idea of the time scale that we're at with the GPUs right now with one work server. And now this is the two slowest ticks, right? So I get a very different intuition from this. The red cross is, again, the crystal structure. The blue dots are actually predictions from this method that the preprint presents. So these are the best weighted ensemble predictions of the weighted ensemble is trying to predict the collective variable is RMSD of the what we call warhead. So that small molecule that would normally be just an inhibitor binding to the target. So you always know that because you always have crystal structures of that to it. You just don't know the protein thing. So you can guide it by the RMSD plus contacts that we pick up from hydrogen deuterium exchange. And that's very nice. In microseconds, you go into what we call atomic resolution, essentially, within below 2 angstroms. So it's good in crystallography. Okay, so from this landscape, it's telling me, yes, the crystal structure is not at the minimum, but it's kind of in that metastable state. And then there's another metastable state on top on the left. It's very far away. Tick 0 describes that. You have none of this information in PC. Again, does it matter? Maybe. It definitely makes cool visualizations. So we're looking here at a movie along Tick 0. Let me actually pause and first explain. So the contacts that are pointed out, what correlates most with going along this tick is there's a contact at the bottom that is going to be broken. There will be this contact in the middle established. And once that happens, we're about at the crystal structure. What the crystal structure is missing is this top contact. And the force field says, no, that is actually further established to fall into the actual middle. I think a big question here is that a force field error or is it actually true? It just you could also describe this process as kind of more hydrophobic collapse. So it might be that the force field just like things collapsing, or it might be the crystal, of this very flexible complex at cryo temperatures just get stuck in this conversion. That's only that Teacup law was only 1 kcal per mole over the global minimum. So, I apologize. Let me play this. Okay, so now it's fallen into these kind of intermediates. They're really like holding on that rope for the proton. Okay, and now we're going towards the crystal structure. That middle contact is established now, bottom's broken. Now we start walking to the global minimum. The top contact established, and we're going to stay there for a long time.
So Russell, for this whole process to work, the ligase has to ubiquitinate the target, right?
Yep.
So like where is the active site of the ligase?
Oh, I don't know actually. Sorry.
Okay. No, that's okay. Because because where it is, you might want really tight binding if the active site is near the target, but you might want looser binding if it's ultimately going to be like an avidity effect where like the ubiquitination actually happens when the target is nearby, but not when it's bound.
See, that's exactly the thinking, right? And you read papers arguing both ways for different systems. So it's clear it's system dependent, and no one knows. There has to be some underlying physical principle, right? But no one.
Yeah. Cool.
All right. So and then you can do your typical MSM stuff. You can k-means clustering and estimate hidden Markov model. And so here you have on the left a 5-state hidden Markov model. We find that crystal structure sits in metastable state 4, which is populated 5%, whereas the global minimum is 63%. On the right, you have your MFPTs. MFPTs are experimentally verifiable, right? So with everything we're doing here, if we're going to use this for drug design, this exercise kind of here, we have to find some quantity that we can see if any of this is real. So again, I leave open the question, and this is the kind of stuff that I like this industrial collaboration to do. I'm telling you what we're thinking, what we're finding useful, and now I'd like to hear back is what are people's thoughts on the usefulness of the message? Should I just do HRMD, which we're talking about hundreds of nanoseconds, and I have my ensemble, or should I really care about kinetics and these TCA plots and making nice movies? And it's not useful because the cost difference is possible to absolutely prohibitive for us. But I do want to get at this question a little bit more of HRMD versus unbiased folding and home simulation. So to looking at here, I apologize, these are not as pretty as before, but I couldn't make the point with the prettiness. These are standard pioneer plots. On the right, these are TCA plots again in the TCA space of the folding and home data set, and so that's on the left, and we project the HRMD data on the right, And so you can see that it's much it's called rough from enhanced sampling. It's definitely missing some, let's call them intermediates there, that you see from folding at home. It appears as if there's just kind of one pathway that HRMD jump over to go from these two metastable states at the bottom to the third on top, whereas folding at home really nicely explores the plateau. And I can tell you, if I cluster these into 100 states, 88 of those are present in HRMD, and the missing 12 correspond to about 15% of population density. Doesn't matter. I know, very hard question. I have to really look at these
even though it's a little like questionable, if you just apply TCA to the HRM data, do you get something that looks kind of similar, or?
You know what? Look at this movie again. I had this idea, right? Because there still has to be some correlation, but like you'll see this thing. It's honestly not the smoothest movie, but like see how it's jumping, right? Like that loop on top is like one frame is there, another is gone. So I think it's just you're gonna pick up something weird probably then.
Okay. Yeah.
But there are methods, theoretical methods actually that I haven't read about, so don't ask me. But apparently you can try to recover kinetics from replica exchange. But so I know that might be worth. Yeah. So back to this. There's the missing states that could be a problem. Maybe not. But the HRMD for a cost that's so we're talking hundreds of nanoseconds, and then we go to milliseconds, right? And so tens of thousands more costly to do unbiased MD. It does explore pretty much the entire TCA. It doesn't fill it in, but I get the idea of what's going on there, and I get these directions. But again, I cannot do TCA with HRMD. I have to have run this unbiased MD to be able to make this plot. A more worrying thing is if I do HRMD and I do PCA and I cluster and I want to get free energies of states, what I looked at is does it misclassify some frames that should be in a high energy state by TCA to a low energy state? Because that's how we always understood this. We go to Payama tutorials where they tell, PCA tells you about things that change a lot and will miss very small changes. So you will not, if you have a difference of one contact between things that are by large the same, and that has a high kcal per mole penalty, you will not pick that up. And that's exactly what happens in this data set. There is a state, if you look at the most populated state by TCA, which is at about 30%. Some of sorry, the other way around. So if you look at the most populated PCA state, some of its frames belong to the most populated TCA state, which is at 30%, but about 12% of them belong to one of the least populated TCA states, which is only at 5%. And the difference is, well, there's many differences, but the most, if you take some samples and you look at the differences in the means between probabilities of contacts, this is the one you pick up. So this is this one contact on the interface. You can see on the left, it's not formed on the right. It's formed is the difference between these two states, right? And again, it doesn't matter. Yes, if you want to propose a mutation, maybe, and you actually want to understand large barriers and change them, yeah. If you want to modulate for user escalators, if you're more interested in just general extraction as this protein over here or on this other patch, maybe the HRMD plus BCA is all you need. All right, so changing topics now to, I've said a lot of things like the simulation for the home is costly and not efficient. So that's the other angle that I'm taking is let's squeeze more out of following home with smart sampling methods. And so I've been doing adaptive sampling for a while now. It is kudos to Joseph of the design of the frameworks. It turned out to be extremely simple to do. All you had to do is use the next gen command again in the project.xml. And so all of this is in this GitHub repo from Redesigned, following home adaptive. And so it has, you see that next state.py, it's a script that gets run on every next gen. And what it does is, I also have now seeds folder, the same way as we have a runs folder. And new seeds are deposited there by whatever modeling that you want to do. And then on next gen, this will go and randomly select one of these seeds to run the next gen instead of continuing the trajectory. So we're obviously losing the continuity of the trajectory now. Every gen is its own thing. And I run a second script, whether on a cluster, if you need GPUs for some bump netting, or on the work server itself, it was just CPUs to generate these seeds, right? And so I've replicated, I think, more of the what they call adaptive goal from Giant Fabrides than Greg's method, because Greg's using RMSD clustering and very, very fine MSMs. I'm doing our TK and just 100 states. You can change any of these parameters kind of approach. And so I have explore only, which is just counts of microstates, and explore and exploit the adaptive goal / fast idea of 50-50 collective variable and counting the states. So nothing fancy there yet. Everyone's welcome. Please do write your scripts. Like to run this. The cool thing is that we can also do experimental guiding of simulation in a similar way. The idea being, rather than scoring by MSM as a collective variable, you score by how well your current ensemble is predicting, be it NMR or HDX data, and then you try to guide it by reseeding to go into where you think it's going to reproduce the experimental data better. So I don't have any results on that yet, but that's not a big part of my job. So maybe next time you hear from me, I'm going to have something exciting about that. And again, everything we develop for at home, the point is we do things that are useful for us, but they're also they'll be useful for anyone else trying to do these things. And our position is that pharma should compete on small molecules, not on computational methods. Really, well, that's right, my position. We're happy to show people how to predict protein complex. We're going to use it for our IP generating stuff on our cluster. But the methods here, the data sets. So the following on data sets is on the Google bucket that is free for everyone to download, 6 milliseconds of data. We also made the HRMD and weight ensemble data. Those we generate proprietary, but we make them available to everyone too. So go. You can ask any questions about the differences in these methods, like I started doing. And so I'd really like someone to pick it up, because it's not. Our goal is this kind of Q-surfing, given free energy landscape, what's going on, not necessarily these fine details of MSMing like I used to do. Make it available, and then in some form, I'd like to tell people what I've learned from my analyses, and then eventually develop better sampling methods, and make the code available to everyone. So that's one guy. All right, thank you all for listening, and please do ask questions.
Thanks, Rafa. I was curious, so have you run this with each of the compounds and like seen what's different?
You mean the protein complexes?
Yeah.
Yes. So we've this is on the trying to stop share. Sorry. This is on the other bucket, the one that we're using for real-time data sharing. We've run all the possibilities. Actually, and there's some more complications, not just the different compounds, but there's two isoforms of either the ligase or the target, I can't remember. And then we also have plenty of data sets from just crystal structures for not just the selection of complexes, but I think we've run all the protein complexes that are available in the PDB. And, you know, that's a good, that's another point of this exercise is we have time, capability to set these things up and run them, and anyone now has a dynamic data set of all the proteins. And so be it an academic or any other company, do whatever you want with it. It's right there.
Okay, cool. Any insight into why, you know, the 3 PROTAC molecules you were thinking about at the beginning have different impact, or haven't dug into the data enough yet?
Yeah, no. You know, I think that the stage that the thinking is at, like, look at the conformations and the length of the linker itself, you know, what that tells you about a conformation. Like, it's oh, by the way, you know, like, I think it's a very desirable skill if anyone's looking for a job to be able to analyze these kind of things. Not many people have thought about MSMs, it turns out, in the rest of the field. And you know, I think if we're gonna get at this question of free energy landscape to design, that's what we're gonna have to do. And oh, by the way, I'll be doing that also for small molecule complexes. So that should be coming in the next few weeks. We're gonna large library of crystals and docked poses. And I'm trying to generate a free energy landscapes of multiple poses, and see what that can tell us about how ligands interact with proteins. And I'm also trying to convince medicinal chemists it might be useful to not just stare at one structure. And that's not easy.
Sounds good. So I want to interrupt briefly and invite the other PIs to switch over to the other Zoom link that Angela sent out so we can discuss some strategy things. But everyone else is welcome to keep discussing science. We have a couple of minutes before that starts if the other PIs have any questions.
Thanks. It was a really nice presentation, Rafa. Thanks.
Thank you.
Okay, well, if there's no other questions, I mean, there's always going to be the happy hours tomorrow at noon Eastern, so everyone should feel free to come on by. And Rafa, if you come by also, people can ping you with questions. I know I'm saving mine for then.
Sounds great.
So I think that's an opportunity as well. So yeah, with that, thanks everyone for coming, and we'll leave it at that.